eating Danone yogurt, and they claim that this lady is 137 years old. Next slide, please. However, we are Ukrainians, and we are already in 21st century, so we thought that we've got our own Carpathians, so we've got our own highland, which is probably the most intriguing and exotic place in the Carpathians is region Hutzulshina, populated by very interesting sub-ethnos, Ukrainian sub-ethnos Hutsuls, and they keep their traditions very well, and especially what concerns food, local homemade food. Nothing can beat their own old ways of making it, and also it's very poorly explored. So we've been exploring Ukrainian highlands in the Hutzulshina region of the Ukrainian Carpathians. And for this search, complex identification and detection of biological properties of yeast with health promoting potential. More than 80 isolates of yeast were isolated from dairy products, mainly from authentic homemade products of Hutzul region. Among them, previously uncharacterized from a microbiological point of view, traditional Hutzul fermented yogurt-like product Guslenka, attracted greatest interest. So you can see the photographs, the photograph of a very famous photograph made in the very beginning of last century, old Hutzul woman, and how this Guslenka looks like. And the bottom left image, it's my own Guslenka. I made it here, so we can do it at home as well. So next slide, please. We isolated these strains from authentic Hutzul dairy, and the results are presented on this rather diagram. And Guslenka is shown in red color. And according to the culture-dependent approach, because we studied here cultivable members of community, microbial communities of dairy products. The dominant yeast for Guslenka were two species, Saccharomycetous cerevisiae and Saccharomycetous marxianus. But we also employed culture-independent approach. We tried preliminary tried generation sequencing using Illumina MySeq, and dominant appeared to be Saccharomycetous cerevisiae, and in such samples, Saccharomycetous marxianus was minor, but still there were dominant species in Guslenka. And talking about the culture-independent approach to understanding bacterial diversity as to lactic acid bacteria, the main two agents of Guslenka fermentation typical for yogurt-like products, Lactobacillus delbrueckii and Streptococcus thermophilus, demonstrated together almost 90% dominance within Guslenka bacterial community. Thank you. Next slide, please. And a couple of words about Saccharomyces as medicine. Like Saccharomycetous can not only promote healthy and active longevity, but also they can treat people from serious diseases. And the brightest star, meet the star, Saccharomyces cerevisiae star, and the brightest star in this area is the world recognized and the most successful in the modern European market. The yeast probiotics, Saccharomyces cerevisiae warburlardi, is a variety of Saccharomyces cerevisiae species. Its effectiveness is clinically proven and is registered in Ukraine and is widely used as a medicinal preparation and row 150. First of all, it is used as diarrhea of any nature during antibiotic therapy to eliminate this bacteriosis, for the treatment of irritable bowel syndrome, and for the prevention and elimination of intestinal inflammatory processes. Next slide. So the beneficial effect of such probiotic yeast on the microorganism is due to its antagonistic and antitoxin properties. Antagonistic, it's suppression of the growth of harmful bacteria and yeasts. Antitoxin, for example, splitting of salmonella toxins by yeast synthesized proteases. Immunostimulating, antidiarrheal, and anti-inflammatory effects due to the large amount of beta-glucans. Yes, Boulardi demonstrated the large amount of beta-glucans and monopolisaccharides in the cell walls, along with antagonistic effect against pathogenic microbes, as well as positive trophic effects. Already mentioned proteins, carbohydrates, amino acids, trace elements, and vitamins. So the health effects of probiotic yeast are trophic, anti-inflammatory, antidiarrheal, immunostimulating, and positive influence with Crohn's disease and ulcerative colitis. But if you can click, please, for me, one click, please. Yes, please, one click. Just one, yes, thank you. I wanted to highlight that we will go down, we will focus on antagonism or antibacterial activity. And now, please, next slide. Yes, thank you. So it's about our research, our modern research that we started with Sulchina and Huslinka. So we tested antagonistic activity of yeast strains isolated from Huslinka, other dairy products, and gastrointestinal tract of healthy Highlanders towards various opportunistic bacteria. We included some Saccharomyces cerevisiae survived for decades in collection, isolated from healthy centenarians and non-Nigerians from Abkhazia. And the results are generalized in this visible component analysis graph. It's just to tell you that, yes, we identified leaders, yeast antagonists with high potential. We found that Bacillus cereus and Pseudomonas aeruginosa strains are the most sensitive to the inhibitory action of yeast. We also used commercial probiotics, Saccharomyces cerevisiae Boulardi, as a control, and it's shown in star symbol as well as the purple arrow. So we found that Boulardi inhibited growth of only these two bacteria and did not show the very best antagonistic performance compared to some other studied strains isolated by us or taken from Abkhaz collection. Thank you. Can we go to the next slide, please? And we selected 18 prospective yeast strains from shown on previous slide of genera Saccharomyces and Cluveromyces to test their ability to inhibit growth of 8 strains of Pseudomonas spp of genus Pseudomonas from collection of Pseudomonas bacteria of Abertay University in Dundee, Scotland, UK. As a result, the best anti-Pseudomonas performance was found for Xenocas as isolate. Cluveromyces marxianus FF8. You can see on the graph the longest columns going down, showing inhibition of all tested, very good inhibition of all tested strains of Pseudomonas. It was followed by isolated from long-lived Highlander, Abkhaz Highlander, Saccharomyces cerevisiae 11K, and also dairy isolate Saccharomyces uvarum FF12 on the right, which was sharing the same cluster with commercial probiotic Saccharomyces cerevisiae Boulardi's control probiotics. strain here. These 3 yeast strains also demonstrated inhibiting effects on aggregative or social behavior of pseudomonads, biofilm formation, the strength of a biofilm, the ability to attach to the surfaces, and also motility, teaching and swimming. These results are still to be published, and the work is in progress. And the next slide, please. And talking about some future perspectives and current interests. Recently, interest in the probiotic potential of yeast and the search for new strains with health improving effect has been growing in the world. Currently, there is an increase in the number of studies of the therapeutic effects of yeast, 1,504 clinical trials only during 2015-2020, including research of anti-cancer potential of yeast, which involve very often glycans. There are also interesting ideas for the use of probiotic yeast in mixed fermentations to increase the health benefits of fermented food products. For example, Italian scientists used Saccharomycetous cerevisiae barbardi for fermentation together with the common brewer's yeast, Saccharomycetous cerevisiae, to produce craft beer with enhanced human health value, reference below. And in turn, it turned out that at the end of fermentation, living cells of Saccharomycetous cerevisiae barbardi were preserved in the obtained beer in the amount of 10 to the 6th power to 10 to the 7th power cells per milliliter. And Boulardia did not spoil the beer aroma and increase the antioxidant activity and polyphenol content of the drink. So you can imagine that you can enjoy your beer now, knowing that you are doing something very good for your health and for your body. And you are having a drink which is much healthier than regular beer, and it's enriched with beta-glucans. What a fantastic picture. Next slide, please. Thank you very much for your attention.
Thank you very much, dear Pani Maryna. And dear participants, please ask the questions. You can write down the questions in chat, or you can just ask.
I would like to ask one question, if it's possible.
Welcome.
Good day. It's interesting because usually, as I know, usually strains of baking yeasts are transferring from one community people to another with the samples of living. And can we, analyzing the phylogeny of yeasts, analyze the places origins of bread baking?
Sometimes yes, but sometimes not, because for example, the regular generally used fungal barcoding ITS region of ribosomal operon, it doesn't recognize some species from sensu strictor. I mean, like 40 years ago, nobody cared. Everybody just called, oh, it's Saccharomycetous cerevisiae, that's all. But now we know, and with genetic markers, we can, but it shouldn't be only ITS region. 18 as the small subunit, ribosomal, it's only on genus level. It's not giving you a good resolution at all for species intraspecific heterogeneity. But there are some, there is a locus B1, B2 in a large subunit, and there are another gene markers, ribosomal RBP2, elongation factor, and there are some other factors and gene markers that can give closer insights where is it from. Yeah. So you mean to follow the geography?
Yes.
Yes, yes. There are even some reviews and some studies. You can look up for some studies where it's a kind of the evolution of these domesticated and non-domesticated Saccharomyces sensu strictor complex strains. Yes, theoretically, it's getting more and more possible with growing success and progress in molecular genetics of yeast.
Thank you. And the second question, if it's possible, bread baking in native people of America. So and strains of yeast which facilitated, are they independent? Do they have independent origins of Europeans? And what the species are there, or it is also Saccharomyces cerevisiae?
So are you talking about Red Indians, some native?
It's a stupid question. I'm sorry.
No, it's not. Nothing is stupid about the Saccharomyces.
Americans didn't know, but were there any fermentations products which were yeast-based Americans of pre-Columbian era?
What do you mean Americas? You mean both continents, northern and south? Yes. There is a difference between fermentation yeast. It's on the third slide. There is a geography. If you go to the third slide of my talk, you can see that it's a wide geography, and it's different species of sensu strictor are very closely related to Cerevisia. But you can see here, not that one. Next. Yes. So, please, back, please.
Yeah.
No. Slide number 3.
Sorry. I'm sorry.
No.
Yeah.
Stop, please. You can see Europe, Japan, West China, Sichuan, Patagonia, Tibet, South America, Australasia. So yeah, and there are some papers. They follow the journey of some species. Because, for example, Boulardia was isolated in modern Vietnam area in the 30s, last century, the 30s, from the fruit skin, because the French doctor observed some severe diarrhea, maybe, I don't remember, it was cholera or some kind of, and people who drank water where they washed some not washed previously exotic fruits, they didn't get ill. And he actually looked in this skin of these fruits. One is lychee, I don't remember the second, but it's on internet everywhere. So this way, this Saccharomyces cerevisiae var. Boulardii was actually found and isolated first, and it was so long journey from this discovery to modern medicine, especially in Europe. Very popular in Europe because it's French doctor, French culture, French strain, French medical preparation. So yeah, definitely, but it's not only Saccharomyces cerevisiae, Saccharomyces sensu strictor are working in bread making, because there are some other, for example, I mentioned Kazakhstania. Kazakhstania is kind of recently renamed, comparatively recently. And before that, anything what is not fully understood was called Candida. So San Francisco bread, I was mentioning, very famous sour bread. It's like a borscht for Ukrainians, for San Francisco is their bread. So it's lactic acid bacteria plus Candida milleri. This Candida milleri now is Kazakhstania humilis. So it's such, I would say, a mess in this taxonomy of fungi and yeast, you wouldn't believe. But now, starting from 2020, it's such a mess because we know too much. With lactic acid bacteria, you wouldn't believe, Lactobacillus now is divided to 25 new species, and you even cannot pronounce some of them. So it's like kind of, it's coming. So we know too much, we are learning too much about the difference between, and it separates some species into different species now. So
thank you a lot. It was an interesting lecture, and thank you for answering.
thank you for attention.
Dear participants, please ask the questions.
There is one question from Julia.
Julia, please.
Yeah, so first I have to thank you for extremely interesting lecture, and actually I have maybe one question about your one of the last slides about the beer. So you actually told that some Italian scientists used Saccharomycetous and Bulgardi for actually fermentation to do kind of healthy beer. So I just wanted to ask that can you say about just an ordinary beer or about non-alcoholic beer? Does it contain this yeast, and can it be like healthy or not?
My point was, and from the very beginning when I learned about Bulgardia, it would be very strange that Bulgardia would be alone on the planet, so good probiotic yeast. So there are Saccharomyces cerevisiae strains which can be as good as Bulgardia and even better than Bulgardia. So about beer, I've been communicating a lot recently with Ukr Pivo, and my understanding, first of all, I started to drink more beer. So a little bit in moderation, because they contain Saccharomyces cerevisiae, Parasborgensis. It's definitely within the sensus stricter complex, but they all have beta-glucans, vitamin B complex. They all have all benefits I showed on one slide. So, and beer, different kinds of beer, filtered, not filtered, not filtered, has more biomass of cerevisiae. Filtered has less biomass, but still they've got some fragments of cell walls, which is rich in trypanomanans, which is great for us. So basically my point is, so beer in moderation, of course, not to get yourself drunk to amnesia, but in moderation is a good thing, which was helping humankind to go through this evolution and history. And as I mentioned before, that when it was very ancient time, it was much healthier. Drink a little of beer or wine, not water, because water cannot, especially in Egypt, in ancient Egypt or Greece, sometimes you cannot guarantee that this water is safe. But after fermentation, after getting some alcohol, ethanol, our ancestors in ancient time, they were drinking alcoholic beverage or kind of low alcoholic beverage. So beer is a low alcoholic beverage. So basically, Bulgardia is an expensive strain. The medical preparation sold in our pharmacies in Ukraine, they are expensive. I doubt that someone is using Bulgardia as a brewery or bakery. It could be because Boulardii should do the same job as the Cerevisia. But if it will be beer with health benefit, it can be much more healthier for consumers. And I get for the market, it's much easier to advertise and sell it. So my answer, I don't know. It was just one paper from Italian scientists. I don't know who else was thinking about making brewery with Boulardii.
Okay, thank you so much for your answer.
You're welcome.
And we have some questions in chat.
I can see Mikhailo. Okay, I just I saw the question. You mentioned that the yeast have some anti-cancer activity. Is there any evidence about this anti-COVID activity as well? This one paper, very recent paper, it's still very early days, but of course it's very beneficial, health beneficial properties of glycans, what is glycomannan complex of cell walls. And I just didn't put it in this presentation, but you can easily find it about COVID. It's number one, anti-cancer as well. Anti-cancer. There are some studies where they're trying to isolate, purify glycans and chemically modify them. And mixes with some drugs as well. And it's a very hot topic now in research. I just didn't explore it for you more because I covered so many things. And so anti-COVID, especially for people who experience not mild form of COVID, quite often there is a complication after you recover with some disturbance of your gut health. So to take some Boulardii might help. You need to consult your doctor, your physicians. So yeah, potential. It's great potential of components of yeast cells or cells either inactivated that or alive like Boulardii, because Boulardii is supposed to use the life. It's a great potential to do anti-cancer and anti-COVID work. That is only the beginning, I guess. And what before that, it was about dough. When you talk about the way to rise it, you didn't mention leaven for bread. Could you explain what is it and what does it consist of? Leaven in Ukrainian is zakvaska. So leaven consists of some nutrients like maybe milk, maybe water with flour. Depends on what kind of bread is, what are you going to bake, and some yeast, some sugar, and if it's lactic acid bacteria, it's some kind of lactic acid bacteria in some environment. So it's a question from Katerina. So did I answer your question, Katerina?
Katerina? I think yes.
So there are different recipes of leaven, depending on what kind of bread and what kind of microorganisms.
And we have from Dmitry one question. Yes, Dmitry?
Can we create more healthy bread with this yeast? Nobody tried. I saw only beer. But well, the beauty and value of Boulardii, it works alive. It goes to our intestines and it it kills pathogenic bacteria. It gives when it's disintegrated in intestines, it gives beta glucans and mannans. So this is a live probiotic, which is kind of, you know, kind of with very high reputation. So, but bread, you know, it should be probably something cheaper because Boulardia is expensive. It should be something, we should find some quite good competing with Boulardia, Saccharomycetous cerevisiae cheaper and make that, because you will have probably glucans. I'm not sure how it chemically will be at such temperatures. So it's probably worth to explore this issue.
Thank you, and you have a lot of thanks in the chat.
I'm very thankful for your attention and interest. I was trying to make you love Saccharomycetous cerevisiae and to get interested in this research, or at least to understand what are you eating and what are you drinking, and be very grateful to our old friends, Saccharomyces yeasts. Thank you very much for the invitation.
It was very interesting lecture with beautiful diagrams and photos. And can I ask you one question, if possible?
Of course, yeah.
There is a lot of information in your slides about the mannane polysaccharides in yeast, which is a therapeutic action. And how do you think could yeasts be applied in treatment of cystitis, for example, when our cystic bladder is infected with E. coli? And I know that there are a lot of medicines containing mannanes can be applied for the treatment of cystitis, but I've never heard about yeast. Maybe it could be some new way of treatment.
From what I understand, we can prevent cystitis by having healthy guts, because cystitis very often appears as a kind of cross-contamination of urinary and GIT systems. So if we've got very healthy microbes, friendly microbes in our guts, healthy microbiome, we will have a chance to get a cystitis. But if it's already on, for example, Boulardia will definitely help if you are taking very harsh antibiotics, antibiotic therapy. So Boulardia is designed to help you to correct your microbiome under this harsh attack of antibiotics, antibacterials. So to put life yeast, no, I wouldn't. If it's mannans obtained from activated cells and purified as a medical preparation, like chemicals, why not?
Thank you. It's also very interesting. Yes, medical application. And thank you very much again for the very beautiful and exciting lecture with a lot of practical advice. Really exciting.
You're very welcome.
Thank you. Beautiful and exciting. Thank you very much, Dr. Maryna. Thank you for your kind agreement to participate in our school.
It was a pleasure for me. I like Odessa. My best kindest regards to Odessa.
Thank you. We have very hot weather today, expecting for thunderstorm in several hours. So
we had it yesterday. We had thunderstorm, and now it's colder, much colder. Yesterday was 33. Yesterday was very hot and stuffy, and then it was thunder and storm, and today is colder, much colder.
hopefully we will have colder
Yes,
weather.
yeah, but the main
Tomorrow.
thing we need victory. We need victory.
Yes,
Any storm will be not an issue for us.
yes, glory for Ukraine. We hope for the
Yes.
victory.
Thank you very much.